Hi, everybody. Welcome to JMBE Live. We do this webinar series to promote and advance undergraduate education scholarship. That's recently been published in the Journal of Microbiology and Biology Education. Today, I'm joined by JMBE's Editor-in-Chief, Stanley Maloy, and our speakers today are going to be Carolina Mehaffy and Grace Borlee from Colorado State. They're going to talk to us about their paper, Upper Level Interdisciplinary Microbiology Cures. That's course-based undergraduate research experiences, increase students' scientific self-efficacy, science identity, and self-assessed skills. Into the chat in just a few moments, I'm going to put a link where you can go read the paper. It's open access, just like all of JMBE's papers, all open access. Okay. Before we get started, I'll remind you of a couple of really exciting opportunities for undergraduate educators that are coming up. One is a webinar series called Getting Started in Biology Education Research. That webinar series will start in about 3 weeks on May 22nd, and I just put a link in the chat to go learn more and get yourself registered. This series is being co-hosted by the ASM and the Society for the Advancement of Biology Education Research, SABR. So take a look at that and consider applying. And then the other thing that's going on that's really exciting coming up is the ASM Conference for Undergraduate Educators. And I'm putting a link in the chat for that as well. And that is an in-person conference in November. And it's going to be in Pittsburgh, Pennsylvania, November 15 to 17. And right now we're soliciting applications for travel awards. And we want content for our conference for submissions of sessions, posters, microbrews. So when you have a chance, go take a look at our website for ASM Conference for Undergraduate Educators and learn more and get your abstracts in no later than June 26th, 5:00 p.m. Eastern Time. So with that, I'm going to turn it over to our moderator today, Stanley Maloy.
Well, welcome everyone. This is another chance we get to actually talk to people who wrote a paper, did the research behind a paper, so that we can ask those kinds of questions that sometimes pop into your head as you're reading a paper. We're going to begin today. They're going to introduce themselves, and then they will take us through their paper. And from that, I'll ask some questions, and we'll take questions from the audience. So if you have any questions, please post them in the Q&A. You can do that anytime. Don't wait till the end. Post them as soon as you get them, but we'll cover them after they're done doing a brief introduction to their paper. So, Grace and Carolina, take it off.
Thank you. Thank you so much. I just wanted to also mention that this was also done in collaboration with other teaching and research faculty in our department, Tracy Kinkle, Bettina Brooklyn, Brad Borlee, and Christy Mayo. And then I will go ahead and quickly, if I can get this go, introduce myself. My name is Carolina Mehaffy. I'm an assistant professor in the Microbiology, Immunology, and Pathology department at Colorado State University here in beautiful Colorado. I teach diversity of cures, and then I also teach a series of undergraduate courses in our Biomedical Sciences program. And I will pass this to Grace.
Thanks. I'm also an assistant professor here in the Department of Microbiology, Immunology, and Pathology here at CSU. I also teach a variety of cure courses. I'm also involved in the graduate program here, teaching in-person courses and in the online Masters of Microbiology and Immunology master's program that we have here at CSU. So let's get started. So we know there's a lot of benefits of undergraduate research. So let's talk about a few of those, right? So when students are able to engage in authentic laboratory experiences, they start to think as a scientist, right? Science identity starts to sort of formulate from late elementary school, middle school, high school. When they get to undergraduate institutions, they really get to do hands-on laboratories, right? So they start to really develop that science identity. Students find research interesting, right? So rather than sitting in a lecture hall, taking notes, right? Once you're doing hands-on activities, there's no time to sleep through a lecture, right? You're actually involved in doing something, right? And you get results and data, and that's really exciting to do. Next off, we know that students that engage in authentic research experiences start to think, where can I take this skill set next, right? For some of them, they might be inclined to explore graduate education, right? Can I get a master's or PhD now that I've already curated a certain skill set? And others think about, how do I take these skill set and apply them to getting a career in STEM? We also know, and data has shown, that getting hands-on laboratory experiences allows us to retain many students in STEM who are from marginalized communities, women as well. We've learned, I think, over, especially during the COVID pandemic, that if we include diversity of voices at the table, we get more ideas on how to solve some of the research questions that are currently we're facing and that we'll face in the future. Okay, so we understand the concept that we need to get students hands-on research experience. So the Vision and Change in Undergraduate Biology Education, A Call for Change, this document is nearly 15 years old now. And we understand, and this has been codified, that yes, getting hands-on research experiences is important for students. And in fact, they state in there, research experiences should be an integral component of biology education for all students, regardless of their major. Okay, so let's think about, can everyone get access to these opportunities? And unfortunately, the answer is not every student is able to participate in authentic laboratory experiences, for instance, in an academic lab setting or in internships, right? So for instance, some students face a lot of financial difficulties, right? So if a lab has an open position, but let's say it's a volunteer position, the student needs to make a decision. Do I volunteer and try to get research experience, or do I try to get a job that can pay for my bills, pay for tuition? Same goes if they're thinking about a research position that's paid. A lot of times that's not paid very well, and they can make a lot more money outside of doing research, right? So these are considerations that we have to deal, that some students have as hurdles in their ability to get hands-on laboratory experience. There are personal reasons as well. Students, for instance, may be really great students, may apply for summer research opportunities, and they may be off campus. But perhaps that student is a primary caregiver, has obligations to their family, and can't leave from their home community that they're at. So this is another limitation that students have to deal with. Simply being aware of these opportunities, right? So maybe a faculty member has a couple of research slots open in their laboratory for undergraduates, but how does that information of the faculty member telling undergraduates, maybe they don't teach those undergrads, how does that information get disseminated to those students? It's not always an easy conduit, and some students may hear about opportunities and others may never hear about those opportunities. Students don't always recognize what are the benefits of getting hands-on research experience. They may understand, yes, I understand I need a 4.0, I need a really good GPA, but in some places it's not really stressed that they need to get that laboratory experience that will give them some other additional benefits, like getting a heads up in grad school or getting a job in the field. Additionally, students are see faculty as being intimidating, right? So if a faculty isn't engaged with undergraduates, students may be afraid to sort of approach them either with an email or talk to them in person. And so that limits some students from approaching faculty to get those experiences. There's also preference for the rising stars in our certain majors, right? So when students do apply for undergraduate research positions, faculty may look and say, oh, this student has a high GPA. This other student maybe is a BC student, and so they'll naturally choose a student that is an A student versus a BC student, right? Although that BC student may actually be more adept and better doing laboratory work. And lastly, I certainly think we need to address this and always be conscious in our roles as educators, is thinking about systemic bias in institutions of higher education, right? This is still a problem for us, and we need to work on dismantling so that every student has equal access to all these opportunities. Our paper is based on CURES. So let's quickly define what a CURE is. So that stands for Course-Based Undergraduate Research Experience. A CURE is an authentic laboratory research experience that's done in the context and the structure of a course, right? So unlike a student that would join an academic lab where they are one person and perhaps mentored by a postdoc or a graduate student, here you have multiple students engaged in doing a project, all for a CURE, all individual projects, but along the same general context. There are 5 elements to a CURE. The first being collaboration. So in a CURE lab, when we have multiple students, in our CURE labs, we can only have a maximum of 12 students. So we have students that will work together, help each other out. If one student is struggling, maybe can't get their PCR work, another student will kind of pitch in and say, hey, let me look through it. Or if you're having problems running a gel, let me help you through that. So it's this idea of being collaborative and sharing data and sharing techniques with each other as they're going through the project. Discovery. This is the best part, I think, about cures for me personally. Discovery is based on the idea that neither the student knows the outcome or the instructor knows the outcome, right? So this element is actually quite interesting. In the cures that I teach, a lot of times the students get very baffled when they ask me a question and my answer is, I don't know, mostly because we're learning what they're getting as data at the same time. So I think that they get that light bulb moment because they've taken so many other laboratory courses that are cookbooks where the instructor knows the outcome of a particular experiment. In cure labs, we don't necessarily know the outcomes. There's a lot of pivoting that happens in cure labs week to week. And per se, I think this is kind of the best part of the cure. Scientific practices. The students are actively engaged in developing hypotheses, testing those hypotheses by conducting experiments, data analysis, and then sort of trying to figure out what to do next with their data that they have gathered. And so this is a really key component of the cure. Iteration. Iteration is the idea that you're repeating things throughout the course of the cure. So that means if your PCR fails, you're repeating, troubleshooting that, right? And you're not necessarily doing experiments once, you're going through to validate that experiment, right? Normally in a laboratory research setting, you're not going to do an N of one and say, hey, that's the answer to everything. In a cure lab, you'll repeat it just like you would in an authentic laboratory experience. Lastly, relevance. What does this project have to do and the tie-in for the students, which allows them that ability to have that ownership and that drive to pursue that project? So for relevance, we have a lower level cure here based on the tiny earth cure. So the students can collect soil, and that soil is sort of their bit of ownership. I've done cures that have tied into faculty members' research projects. We can also think about cures that are related to the community and having tie-ins from the community into cures as well. So that is what we have for cures.
Okay, I'm going to now walk you through the theoretical framework of our study, which is based on the tripartite model of social influence, which was initially developed by Kelman in 1956 and then refined later on in 2006. But then it was Estrada and co-authors in 2011 that used this framework in the context of experiences of undergrad students, specifically in STEM. So I'm going to show you there are three main constructs or characteristics of this model, which includes self-efficacy, and I'll explain each of these in a minute, sense of belonging, and value orientation. All of these three are interconnected to each other, and several authors have reported that these construct or these metrics have a positive impact in students' experiences. And they have also shown that specifically looking at research experiences during summer internship or summer research experiences, as well as research experiences in working with an independent research lab, increase these metrics, and that helps the students persist in STEM. And also they have shown that specifically sense of belonging and value orientation, which I will explain in a minute, are directly related to students pursuing a career in STEM. So we wanted to see how this model could be used to study the benefits of cures in our students. So I'm going to start with self-efficacy. Self-efficacy is the idea that as we are researchers and the students come in this research environment, there are certain type of conduct behaviors or certain ways that we are expected to behave as researchers. And behaving as researchers will be then be seen as something good, something that can be rewarded. In the context of cures specifically, that self-efficacy can be seen as students or can be impacted as students complete research. They also have data ownership In all of these cures, we see, well, we'll explain you some of the cures that we included in the study, how the students really have ownership of their own data and how their data is different from their peers. And then again, scientific skills. The other construct is sense of belonging, which is directly related to scientific identity. And this is just the idea that, do you believe that you are a scientist? Do you feel and see yourself as a scientist? Of course, within the context of research experiences, regardless of what that is, if you are doing research, you have a higher chance that you will see yourself as a scientist. Specifically within the cures, but again, this is more general to research experiences in general, there is that sense of community building. So you're not only seeing yourself as a scientist, but you can also see that other people can perceive you as a scientist. And then you also are building and collaborating in research. The other aspect, which goes back to those tenets that Grace mentioned of what a cure is experiencing discovery, something that we don't really know. Even the instructor doesn't know what the outcome of the project is going to be. And then finally, value orientation. This is the idea that we can see science and research as something that benefits society. So is that tied within how is that our findings will ultimately benefit my community? In this case, what we see with the cures is that we're connecting and the students are able to experience the connection of scientific research with societal values and advancements in society. Okay. Within that, then, the goals of the study were to identify if self-efficacy, science identity, and value orientation are positively impacted in the students that are completing our upper level cures. And then we also wanted to know if students are increasing their confidence in specific skills. Okay, so for our study design, we decided to use several different cures and collect the data in different cures that we taught throughout the years. Our setting is an upper level cure program. So here we're talking about mostly juniors and seniors, students that can register for this class. Although we do have a few other students at other levels. This is an elective course in our Microbiology and Infectious Disease concentration, which is part of the Biomedical Sciences program. It is a 4-credit course, 5 hours a week, semester-long course. In addition to all the tenets that a cure has, as explained by Grace, we also, in our cures, mimic or try to mimic what the students will experience in an independent research lab. So students are expected to be involved in all aspects of the research, from collecting data and analyzing data all the way to autoclaving trash and making sure the lab is clean and tidy for everybody. Okay, so in terms of our methods, we collected 3 different types of surveys, pre and post. So we collected the pre-survey on the first week of classes and the post-survey in our last week of, or last day of the lab. We collected the Teams survey, which relates to those 3 main constructs that I just explained. We also collected a questionnaire on self-reported skills that included skills in microbiology, molecular biology, bioinformatics, research and science communication. And we also included a cure survey, which was modified from other authors, in addition to collecting demographics of our students. And this was all approved by our IRB. Okay, so from here, I just wanted to show you that the cure demographics of our students, we're not showing the CSU at large or our department at large, but it is very similar to what we would see here, with a predominantly white population of students, followed by students that identify as Hispanic Latino. And as I mentioned, you'll see mostly juniors and seniors in our cures, and we have some transfer students as well. Okay, so one of the reasons why we decided to study the program as a whole instead of looking at specific cures is because, well, two reasons. One is because our number of students in each cure is small. Our cap is 12 students because of our cure lab, that is the capacity, but also having a small course really help us as instructors be able to personalize the research experience a little bit better. We are able to help students that may come with different skill levels and still guide them throughout the entire project. And the other thing that I wanted to point out and why we decided to do this as a program is because our cure program is quite unique in the sense that we do not repeat the same cure every semester, but rather we are trying to bring innovation, and we try to do a different project as much as possible. So we have 5 different cures that we included in this study, and I will walk you starting with the first one. The first cure one, which I led this cure, was called non-tuberculous mycobacteria and climate change. And I'm a microbiologist by training, mostly doing tuberculosis work and research, but I'm also interested in non-tuberculous mycobacteria. These mycobacteria, we know they're environmental mycobacteria. They're found in soil and water. And in the last 2 decades or so, we've seen an increase on infections due to these opportunistic pathogens. So one of the reasons that has been kind of like thrown out there is that perhaps the climate change that we're experiencing can change the biodiversity of bacteria in general, and specifically of non-tuberculous mycobacteria. So in this cure, the students learned first kind of like that connection between human health and environmental opportunistic infections. They collected samples twice in the semester to map 2 different seasons, so we could have some climate data. And then the students follow with culturing their samples, looking specifically for non-tuberculous mycobacteria, but also other bacteria in general. And then they also did complement the research with 16S next generation sequencing, all the way from extracting the DNA from soil and water samples to creating their libraries, to doing the data analysis in Galaxy, to getting these are called chrono charts that are interactive, and the students can kind of like click on the different components and then look at the composition of the taxa in their sample, and then also compare the different samples that they took. And I will pass this to Grace to talk about the other cures.
All right, so I was the lead for Cure 2. Cure 2, I had help from Drs. Brad Borlee and Dr. Christy Mayo as well. This project really ties into the research that Dr. Christy Mayo's laboratory does. She's particularly interested in the Chelicoides sonorensis biting midge. Some of you may refer to these as noceums. These arthropods feed on ruminants. They take blood meals on these ruminants. They're relevant because they vector a number of agriculturally important viruses, including blue tongue virus. And I'm interested as a researcher in arthropod microbiomes, generally speaking. So what we did is we had Chelicoides sonorensis biting midges that we had them feed on blood meals that were spiked with various antibiotics or NSAIDs that they may see if they were feeding on cattle at a feedlot. And the students then processed these culturable bacteria and isolated pure colonies, and then tested their pure colonies in quorum sensing, quorum quenching assays, antifungal assays. They also look for antibiotic producers using the escape pathogens from the Tiny Earth series. At the end of the semester, the students took their pure isolates, and then we ID'd the bacteria using a MALDI-TOF at the veterinary teaching hospital here at Colorado State. So they got exposed to sort of high-tech bacterial identification technology. While we were working with culturables, the students isolated total DNA from their samples, and these DNA were PCR and library prep for 16S microbiome analysis. I will give a shout out to the webinar series Microbiomes for All, which is where I learned about the pipeline Nepheli. If you're interested in having your students exposed to 16S data but without needing to code and learn Python, this is a really great way where you can have basically input of reads, and then all you need is a metadata file. So what that allows the students to do is generate relative abundance plots and then sort of look at what they've found from culturable data and compare that to the unculturable in their sample. Cure 3 was done by Dr. Tracy Kinkle, a co-author on this study. So they were interested in looking at Alaskan red foxes and the parasitic infections that they carried. So they specifically looked at the microbiomes of the nematode infections of these red foxes, also did fecal floats with these parasites as well. Cure 4 is one that I co-led with Dr. Bettina Brockling, who's also here at CSU. This was really taking advantage of wastewater samples that were being collected throughout the state of Colorado with Dr. Carol Willis. She's another professor here at Colorado State University. So she was collecting them to basically test wastewater samples for the presence of SARS-CoV-2. What will happen is that we had these wastewater samples, and then the students generated primers to do our RT-qPCR and either run them on the RT-qPCR machine or digital droplet PCR for detection of either bacterial or viral pathogens. So they could pick samples from Colorado State, they could pick samples from the general Fort Collins area or even Denver to analyze and see if those pathogens were present in the wastewater samples.
Okay, and then our last cure that I want to show you here it was a very unique cure because I led this one, and it came out as an opportunity that I had to be trained in this cure by a group at UC Davis led by Justin Siegel and Ashley Batter. That is an NSF-sponsored program where they train instructors into the specific the cure, and then the instructor comes into the institution and implements it. In this cure, the students have experience with protein modeling, looking at specific mutations that they select, and then come up with hypotheses of what the specific mutation may have, how they may impact the enzymatic activity of this protein, which is the enzyme beta-galactosidase. Once they have their hypothesis, the students will do site-directed mutagenesis to introduce the specific mutation that they want to test, followed by cloning, and then confirmation by Sanger sequencing that the mutation is there. And then once that is done, then they will proceed with protein extraction using E. coli and then enzymatic activity. One very nice thing about this program is that the students also are able to contribute to a database that is managed by UC Davis, where many different students that are throughout the nation doing this cure can add their data to it. Okay, so now let's move on in some of the interesting data that we have of this study. And before I walk you through some of our results, I want to show you and to introduce you to this type, just in case if you're not familiar with this type of graphs. These are called violin plots. And then what you would see in the actual plot is in all the plots that we have, since we collected pre and post data, then you will see data for pre and post. You will see in the middle of each of the plot is the median. In this case, the median corresponds to the median average score for each of the metrics that we are measuring. This is a Likert scale data for all of the measures that we did. And then we'll see the upper and lower quartiles. The nice thing about the violin plot is that we not only see kind of like where the average or where the median is, but we also see the distribution of our data. For example, you see that in this case, most of our responses fall around the median, as may be expected. And then we can see that only a few responses really went kind of like outliers, either very high or very low, above and below those quartiles. The other thing that I wanted to mention here is that you will see here, I put in the scale that we were using, which is different for each of these metrics. For example, for our self-efficacy scale, it goes from 1, not at all confident, to 5, absolutely confident. And this, as I said, is the average, because in each of these surveys, we have 4 or 5 different statements that the students respond to it. Okay, so now for the actual results. In here, I'm showing you the results for our self-efficacy data. showing that after the cures, the students significantly increase their sense of self-efficacy. Here we have our science identity, which is related to sense of belonging. And we have here from strongly disagree to strongly agree. And then we can see here how, first of all, the distribution in our pre-survey, it is a lot wider range here. And then in our post-survey, it not only increases, but it's also the distribution is more narrow. So we see that our students increase their science identity after these cures. And then finally, and this was something interesting, is that the scientific value orientation, we really didn't see any change. But what is interesting is that, and then as you can see here, the scale is a little bit different because we wanted to show you the distribution. And as you can see here, most of our responses fell between 5 and 4, which starts with 5, very much like me statements. So what this indicates is that our students are already coming into the program with a very high sense of this scientific value orientation, which again, this speaks to the idea that the students appreciate science and research and their benefit and how it benefits society. So what we think is that the students that are already feeling that sense of, yes, research and science is really important for society are the students that may be self-selecting and enrolling in these cures. Okay, so that was for our TIMSI data. For the competencies, as I mentioned at the introduction with our surveys, we created a questionnaire that would ask students to kind of like rate themselves from no experience or knowledge to high level of expertise of knowledge in different fields: molecular biology, microbiology, bioinformatics, research, and science communication. Just to give you an example of what the type of questions that we would ask, for example, for molecular biology, we would say something like, I have experience doing a PCR, right? So the students would say, no, I don't, or would rate themselves from 1 to 10. And then each of these categories will have multiple questions. So you will see again here is the average of those. And then what you can see here is that for each of these categories, we see a significantly increase in students' confidence in saying, yes, I have expertise or knowledge in this particular technique or this particular field. The other thing that you can see here, which we found interesting from the perspective of our own program, is that students come with high levels of confidence in microbiology techniques. But then when we have molecular and bioinformatics, we see a much bigger range. And then what is nice about these two is that our CURE program is filling this gap because we are bringing new technologies, we're bringing state-of-the-art technologies to our program. And then finally, even though our numbers are small in terms of our students that identify with a race or ethnicity that has been historically marginalized in STEM, we still wanted to see if with the numbers that we have, we could see a trend or a pattern of our students that have a racially minoritized status. And then what we see is that even with these small numbers, we see an increase in self-efficacy and science identity in our students from these backgrounds. And then as expected and similar to what we saw in our other data compiling all of our students, there is no difference between the scientific value orientation pre and post. And then with this, I'll pass Grace to final points.
All right, so just some conclusions from our study. So our study and many others have shown that cures positively contribute to students' self-efficacy and science identity. Although we've seen what others have shown about not such a higher increase in science community value orientation, right? So what we're thinking about here is in our program, the way it's structured, we just talked to you about upper level cures, which are predominantly taken by juniors and seniors. We also offer a second semester for first year students in our concentration based on the Tiny Earth model, right? So we're thinking about filling in those gaps with 200 and 300 level cures courses to sort of have students get additional benefits from those cures between their first year and fourth years. Using self-assessment, students gained confidence in a number of techniques, particularly bioinformatics and molecular biology skills. And what we also want to show in the study is that participation in a cure is key. So it's not based on the instructor, it's not based on the content or the subject matter that was taught, but simply participation and being active in a cure really does students reap a lot of benefits from that. So future directions, what are we thinking? We're thinking about additional interventions in cures that can kind of raise those skill scores in science self-efficacy and science identity. What else can we do to sort of bring those metrics up higher, including in marginalized communities as well? As I just mentioned, talking about bridging our cure curriculum as well, right? We offer them at the higher levels and one at the lower level, but try to bridge that between 200 and 300 level courses. Also thinking about interventions is how can we have greater tie-ins with our communities, right? So we do a lot of our work sort of based on interests of the instructor. Some of it can be tied into faculty research as well, but thinking about projects that may engage our Fort Collins community. And lastly, what I want to say, and I'll speak for Carolina here, we both really enjoy doing cures. They are quite a bit of work, but honestly, I think they are incredibly rewarding. So if you're on the fence whether or not you'd like to engage in doing a cure, I say go for it. I just completed a CRISPR cure lab, and honestly, I think it's the most fun I've ever had teaching it all. So if you're on the fence, you're always welcome to reach out to us. We're happy to help anyone to engage in learning about cure courses. With that, we will take any questions, comments, and feedback from folks.
Thank you very much. That was really very nice. So Young by Kim had a three-part question. I'm going to break it down individually so you can answer each part separately. And the first one, I think, gets down to the crux of when you look at the outcomes of your study, how does that compare with the many other studies on cures that have been done with different populations and different types of courses?
Yes, I can speak specifically to the TIMSI data because that is what a survey that has been done, but the number of studies that have used that survey is very small. Like I can count them with my hand. Specifically when it comes to to life sciences and related fields. What we saw, it was very similar to the other studies. High self-efficacy, high science identity after the cures, while the scientific value orientation, no significantly different. Many of these studies were done similar to us in a setting where the study was, or the cures were elective cures, which goes back to the idea that idea of self-selection. So I guess I stop there, and then if there is any follow-up question, I'll go.
Let me just
do you want to say something?
So sort of the idea is that we also know anecdotally, and we mentioned this in the paper, that a number of our students that are enrolled in our elective cures are involved in faculty research programs as well. So they're double dipping between the experience of getting it in the academic lab, but also in a structured course setting as well. So again, we're sort of enriching for that population.
True.
So let me follow up on that because that was one of the questions I had. So yes. I wonder if the difference that you see in these different criteria over the course of the cure, if that's partly from the cure and partly from their other research, the traditional research experience, if you would.
It is.
How can you separate those two?
Yes, it is possible. We kind of like we noticed this early on, maybe when we had completed the first 2 or 3 cures, and we decided to add another question to the survey, but then we didn't really include it in the study because it wasn't done in every single cure. And then basically we asked the students, do you have experience with research? And then one of the questions that is also in the survey is, do you see yourself doing research, right? But then there was really, we didn't see any pattern in terms of the students seeing themselves doing research or not based on their responses. And then what we know is that probably 50% of our students are doing research in other labs. 50% they come in their senior year, and this is the last chance they have to experience research. And just, I guess, basically just based on our observations in the class, these students love the experience. They come in and they get a lot out of it because they come right at the end of the senior year. They may already have made their minds of what they want to do as a career. But I think this opens their eyes for the students to be like, okay, the research is definitely a possibility.
So Kim asked another question. It's not clear to me that you can answer it based on your paper. But if you take any course, so let's say you take students in a traditional laboratory course, and you applied these criteria, would you see a gain in that? And likewise, if you have somebody in the traditional research setting, right? So if you could do a comparison across these different groups, is the relative change similar or different in those different settings?
Do you know studies?
Well, this study by Estrada and co-authors, they really were the ones that developed this TIMSI within the context of undergraduate research. They didn't do any cures, they didn't apply this to cures. They were looking at students that have just any undergraduate research experience, either working, I believe, in a lab or as part of a summer research experience, and they see similar results. There is an increase in self-efficacy and science identity as well.
So there are a couple of other questions that have to do with implementation, if you would. And that is, Jacob asks, what was the hardest part of implementation of cures? And Vida added, how do you fund them given how tight departmental budgets are nowadays? So both those have to do with how you can do it, how you can put on a cure.
I think I'll give this to you too, but just I think the first thing is to just to want to do it, of course, right? Imagine if you're asking these questions because you really want to do it, and then we get very creative in terms of where we get supplies or where we get maybe some equipment. We have built a really nice lab where we have equipment where we don't have to figure out as much in terms of where are we going to do this. And then we also have a very nice support from our department so that they do allow us to have a special budget for our cures. We still have to be very creative and very strategic as to what is that we're going to do. And I know Grace can also talk about some other partnerships that she's done to be able to get maybe a better budget.
Yeah, I mean, cures are expensive to run. They're not cookie cutters. The way we've been able to sort of stretch our dollars, if you will, when labs do their giveaway piles or giving away piles, we go through and collect things as much as we can, even if we don't have the intent of using them, because we never run the same cures twice. We're weird that we change them up every semester. I've recently partnered, this previous cure that I just concluded was a CRISPR cure. I partnered with an animal health biotech company in town, Zoetis, which used to be part of Pfizer Animal Health. So buying the sgRNAs, which were very expensive per each student, they were gracious enough to pay for all those. So kind of if you can partner up with someone. Also for my 16S sequencing runs, I did not buy supplies for any of the library prep. I had a colleague that was doing 16S microbiome runs, and when they had empty space on 96-well plates, we would jump in with our samples and they would sort of max out their plates. So being thriftiness, taking advantage of the resources at your home institution and taking advantage anytime anyone said free lab equipment or free supplies. We were definitely good at that. So yeah, it is stretching a dollar, and certainly doing ones that are bacterial based on using bacteria is cheaper than certainly eukaryotic cell culture. So being very thrifty and thinking about maximizing every dollar that we have.
So I'm going to jump into another question. That is, one of the things that you mentioned in the paper is that there's a difference associated with gender, right? Where women show a bigger increase than the male students. And I wonder if you could say something about that. Is that because the initial self-confidence started lower and it increased to the same level, or what?
To be honest, I was a little baffled by that result, and we believe that we need to be able to continue collecting data to be able to understand a little bit better why that is. We have our population slightly higher for students that identify as female versus those that identify as male. So to be honest with you, I don't really understand why that is. Do you have any other thoughts? I think we were both kind of like a little baffled about that specific finding. Yes.
It would be a very interesting question to pursue further.
Follow-up study. Yes.
One of the other questions I had was your third criteria, scientific community values, is an interesting criteria that my understanding is you could think of it in different ways. You could think of it as how scientists view their impact on the community or acceptance by the community. But you could also say, within science, do we have community values that we hold as being real key, right? As part of the culture. And the two things I think about a lot when I think about that scientific culture, one of them is responsible conduct of research, and the other is laboratory safety. They're both ethical issues, right? And so I wonder if you have any, when you ask the questions, if you probed some of those science culture issues when you were looking at this question.
You want to go with this one?
It's yours.
Okay. I believe that I'm trying to remember exactly the survey. There are 4 different statements, but they are very much geared towards appreciation of science and research for the benefit of the community. So I think it is more towards that side of things than looking at potential ethical issues or conduct of research. Yeah.
We do, for all of our cure courses, though, in terms of biosafety, we do have them go through all the we have a biosafety officer come and kind of give them a rundown for a full class period. So we do incorporate that already. But again, some of them may be coming in with that information from their lab setting. So they may already be bringing in that information and they're just hearing it for a second time. But yeah, we do have that as a critical component of all our cures.
Just to clarify, there's a difference between knowing about them and having it part of your culture or part of the scientific culture.
Yes, true.
So that's the cultural aspect that I was getting to, which I think fits into that third criterion there. Right? So, and it's really important. I hope you could tap into that question as you move on in the future.
Yes. And then we, if I can maybe talk a little bit more about that. We also talk with students the importance about, so for all of our students, instead of regular lab reports that they may do in other labs, we have them keep a notebook like you would do in a research lab. And then we really talk about the importance of being very thorough with their notebook. Even if they have negative data, even if their data is not what they were expecting, it still has to go in their notebook. You have to be very accurate and very specific and detailed about what the protocol was, any deviations of the protocol, and we really emphasize the importance of reproducibility in science, and how anybody that comes in, checks that notebook, should be able to reproduce what they did. So.
Thank you. So Jacob asked another question, and it was, have you thought about other theoretical frameworks for this study in addition to the TIMSI?
Do you want to talk about intergenerational learning?
Other frameworks, I think, for analyzing.
Well, I guess the fast answer is no. We're really because this framework has already been validated. And then also within the main emphasis of identifying ways that students persist in the program, and also identifying the important metrics for students that once they finish the program, how are they going to continue with a career in STEM? And then the Team C model is the one that has been out there looking specifically at these questions. And that's one of the things that we're most interested about as researchers and educators, is how do we help our students be successful, but also how do we help them to stay within the program and continue doing research once they leave the university. Yeah.
So you made a very important point when you did your introduction, and that is the fact that cures, as well as a traditional research experience for undergraduates, really help with persistence, and they really help, especially minoritized students, in terms of moving forward. But then the question is, and you also point out that the research experiences are very limited, and very often your CB students can't access those experiences. But when you have courses when you have the numbers that you have, are you really reaching out to the number of students who really need or would benefit from this? And it depends on the size of your program. So that's it's not a critical question, it's a genuine question.
No, that is a good one. And it's one of the things that we are constantly thinking, because we know, based also supported by our data, that these electives are self-selected. So the students that enroll in these electives are really self-selecting. They may actually probably don't need any additional research. They already know the importance. They already feel that they belong. So something that we're very, something that we really want to do is to be able to have require courses that are cures, have cures that are integrated within all the labs in the program would be ideal, because then the students not only have that one semester experience, but then it's something that they will be able to do throughout their program. Have sequential cures, for example, would be something that we are trying to incorporate into our program, because I totally agree with you that the students that probably need this the most are not the ones that are getting enrolled in this. Do you want to say something else?
No, we just certainly don't take that as a criticism. It is a critique of our own program. And we realize that, yes, as Carolina mentioned, a lot of our students are self-selected by that point. And yes, that is, they get certainly benefits out of it, but are there students who could have gotten greater benefits out of it and didn't enroll? We think for sure we are missing a large part of our population that we're not reaching out to and not engaging. And so we need to retool how we have students enroll in these cure courses and have more selections or more offerings so they can be enrolled in them. Because yes, it's been shown by many others that there are benefits to these cures, but only if they're enrolled. So we have that gap right there. So thank you for the question.
Well, we're getting to the end of our time, but I want to say I really enjoyed your paper. And I think that this question about the benefits of cures and the number of students who can access them is a really important question for all of us who in education. And the one other thing I'd say is, I think the points that you make about the skills that the students get from the cure, we want all of our students to have those skills.
Yes, exactly.
And so,
agree. Yep.
So thank you so much. I really enjoyed your presentation and your paper.
Thank you.
Thank you.
Rachel, back to you.
Thank you, Stanley. And thank you to our speakers. They diligently worked hard to bring you their presentation today and make it accessible to a wide diversity of audience. And we do appreciate their time sharing their scholarship with you. I'll do a couple of closing announcements. So I hope I'm sharing my screen. So we have some really exciting events coming up for undergraduate educators. We have our in-person ASM conference for undergraduate educators, November 15 to 17 in Pittsburgh. Our registration is not up on our website yet, but The registration deadline for the early bird discount will be September 16th. Website is www.asm.org/ASMCUE. Don't forget, submit your abstracts by June 26th. Travel awards will be due April 12th. Also, this is exciting. This is the first time we've ever done this. We're going to offer mini ASMQ, and this will be in conjunction with the ASM micro meeting on Monday, June 17th. And due to a special arrangement, we're offering this event free, and it's in-person only event at the Georgia World Congress Center. It'll be 9:30 to 2 p.m., and we're going to engage the local Atlanta community. So if you live in Georgia or Metro Atlanta, do go register yourself and meet new friends and learn some new teaching techniques. You can register at tinyurl.com/mini-ASMCUE, and do make sure that you put the URL that I just told you is case sensitive. So do capitalize the M and the ASMCUE. In addition, we have our upcoming webinar series, Getting Started in Biology Education Research. And that's going to be really, really exciting webinar series. And Stanley is one of our speakers. And I'll put the link in the chat. You can go get yourself applied right now. And if you love this kind of talk today, you're going to love learning the techniques of being an education researcher. You'll gain fundamental skills. And then if you take this webinar series, you can join us for an in-person workshop to help you further develop your skills and develop research questions for your own institution, your own context, with an in-person workshop at ASMQ. So take a look at all these exciting opportunities. Don't forget, our next free JMBE Live webinar will be Friday, May 17th, 2:30 p.m. Eastern Time. You'll have to register yourself to get on the list. And the recording of this webinar and all JMBE Live webinars are available at our YouTube playlist. Thanks for attending, and we hope to see you at a future event.